Thank you. The floor is yours, Flavia. Thank you.
Okay, thank you very much. Thank you for the invitation, Peter, and all of you for being here. It's my pleasure to present in this seminar series that I didn't know about, and I'm very happy to be here. So, as the title does not say, I'm going to talk about evolution and phylogenetic trees and the signatures that we can detect in these phylogenetic trees. And for this, I've been using a model, and I'm going to discuss under this framework of the model that I'm using. I'm going to discuss different results that we got, and then we are going to. I'm going to present in the end about how games can affect the evolution process and how it changes. What are the signatures left in the phylogenetic trees? And just to start, I want to thank all the institutions I'm attached with at some point of my life and all the fellowships that I got so far. I hope I get more. And all the friends and colleagues and students that I've worked in these works that I'm presenting now. And there are many more. Fortunately, I'm very happy to have so many collaborators of other works that I'm not presenting today. And I do believe that collaboration is the most interesting, important thing in science. So this is why this is the first slide. So thank you very much, all of you that worked with me on this life. So I don't know if you know, but my background, I'm first a biologist and then a mathematician. And because I'm a biologist, I'm very, very interested in diversification. And maybe this is one of the main questions in biology or in evolution, how the species form and how they coexist in nature. So because this is one of the most important questions in biology, this is also my curiosity. And we want to understand why we have so many different species and what are the evolutionary patterns between these species. I mean, how these species are related to each other, how they evolved from each other, who came first, what are the ancestors of these species. We want to understand their evolutionary history. And of course, if we want to understand the patterns, we want to understand what are the shaping processes that are leading to these evolutionary patterns. For instance, we want to understand, in this case, that is not my work, but it's a very beautiful work. We want to understand, for instance, if space is an important force to form different species, if they coexist under the same area, or if they are very splitted in different regions. And when we talk about evolutionary patterns, we are talking about macroevolutionary patterns, for instance, the phylogenetic trees that I presented here. And when we're talking about shaping process, one of the shaping processes that I'm interested in are the microevolutionary forces. For instance, mutations, for instance, the use of the space, for instance, how individuals reproduce with each other. So my work and the work of the group that I'm inserted in, we usually work in what we call the macro-micro gap. So we want to make the bridge between what is happening between individuals inside individuals level and understand what is happening in the species level. And the diversification process is very well studied in many cases. And one of the most well accepted things that make species to split in different groups of individuals that cannot reproduce anymore with each other is the geographic configuration. For instance, this one where you impose some kind of barrier, individuals, they are split in different areas and then they accumulate mutations, they accumulate differences, and in the end, they form different groups of individuals that don't recognize each other anymore as individuals that they can reproduce with. So that's what we call, under one of the many definitions of species, we call these different groups as species. And when we have this kind of barrier, this is called allopatric speciation, because there was something that made these things accumulate differences in the space, and then it's called allopatric speciation. And this is one of the most well-accepted forms for speciation. It's the one that made chimpanzees and bonobos to split in different species. The Congo River was the barrier between these apes. But there are other kinds of speciation modes, and the other one, not that well accepted, many times with very few empiric evidences, it's the sympatric speciation. During the sympatric speciation, individuals are using the same space, they have access to the same individuals, but somehow they split into different groups of individuals that don't recognize each other and then form species. And this is likely the process that led to different species in African lakes and Central American lakes of the cichlid fishes. This is like the main empirical evidence for sympatric speciation, but there are other. And this is one of the things that we want to understand, how individuals that have access to each other, can reproduce to each other, can form different species, how the genetic configuration breaks, how the genetic flow breaks between these individuals, forming these species. And to make this, we work on this framework that was proposed by Derrida Hicks. Derrida and Hicks, they are two physicists that were working on this very theoretical work of speciation, and they proposed this kind of model where individuals are defined by a biallelic genome, and in their case, it was an infinite genome. And they worked with simulations, working on the genetic similarity between these individuals, but here, and because we want also to understand what happens in more like real cases, we work with finite genomes. So we want to understand how individuals that are defined by finite genomes can form different species. And then we represent each individual by this vector of biallelic genes. And these biallelic genes, we have like B genes, the length of B genes. And the individuals are aploid individuals, and they reproduce sexually. And just to understand better how they reproduce, I'm going to explain here slowly, but this is going to be used in every work I'm presenting here. So an individual that we call a focal individual looks for a sexual partner to reproduce. And it's going to reproduce with those individuals that have some compatible genetic distance. So if we compare the genome of the focal and the sexual individual, we can count the differences by what we call the Hamming distance, and see if this difference is low enough or smaller enough for them to reproduce. And the offspring, if this genetic distance is it's smaller enough for them to reproduce, they're going to give rise to an offspring, and this offspring is going to inherit the gene from the focal or of the sexual partner with 50% of probability. So here, in the first position, doesn't matter where the gene comes from, but the second position here, we can say that it came from the focal part, for the focal parent. And then on the fourth position, it came from the sexual partner. So each loci here is inherited independently. And we also have a mutation rate, mu, where each of these positions can mutate with a given probability. And we do this individual-based model in a spatial explicit framework. So we have a lattice of L by L. It can have a different shape, but here we are working on this square shape. And the individuals are placed in this space, and they reproduce with those individuals around their neighborhood. So one individual here, the focal one, it can choose the partner inside this neighborhood. And once they are inside the neighborhood, the partner must be compatible, must be lower than a given distance G. And we can also use a larger mating range. We can look for the partner here, the focal individual is here. We can look for the partner everywhere in this, mostly everywhere in this lattice. So we can vary the mating range or the home range these individuals have. And when we do this, we go from this very similar to allopatric, but it's usually called parapatric, to the sympatric mode of speciation. Is there any question so far? No. Okay, I will continue. And we also need to define species. And the definition, we have many definitions of species in the literature. And here we are using one of them. So if we link individuals by their, those individuals that can reproduce with each other. So if we link the individuals which the genetic distance is smaller than the value G. If we are linking them, we can see that some individuals can reproduce with each other, but other individuals like this one and this one, they cannot reproduce with each other. But somehow there is a gene flow between this one and this one through a third one. So if there is genetic flow, we say they belong to the same species. In our model, as time passes and we have mutations, we have sexual reproduction, what happens is that this genetic flow is broken at some point, and individuals will be splitted in different groups of individuals that cannot reproduce with each other. So this group here, this gray group here, there is no individual that is linked to the yellow one. So we say here that these are different species. At some point, this other group here will will have no genetic flow with the yellow one anymore. So there is a third species in this case. So basically, we are defining species by the genetic flow. And what we do in our model, we start usually with a homogeneous population. Everyone is equal to everyone else. Like we start with every individuals with 0, 0, 0, 0, 0, like the genome is only 0. And as time passes and they reproduce and mutations happen, what we observe is that we form a species between these individuals, among these individuals. And something we wanted to understand better was, there's Victor raised a hand. Sorry, Victor, you can say.
Thank you for that. I was just wondering if how do you choose the number of sexual partners you have, like the number of offspring an individual you have?
Everyone has one offspring.
And then the parent die, or is there overlapping generations?
There is no overlapping generation. So yeah, the parents die.
Okay.
Okay.
Can I also ask a question? I was just, how do you model the event that, or when do your model individuals, when are they unable to mate with each other or reproduce sexually with each other? Is that one particular trait or loci? So
Yeah, so what we do is that every individual has the chance to reproduce. If this individual doesn't find a sexual partner in their neighborhood, in its neighborhood, it's not going to reproduce. It's not leaving any offspring. And then we give the chance of another individual to reproduce twice.
But that, I thought that the chance of finding a mate was just this radius. So as long as there is an agent in my radius, I can have that as another part.
Yeah, so usually when I think it will be more clear after, but usually when the radius is smaller, it's easier to find a partner.
Okay, well,
Then,
I will wait and I will probably understand later.
yeah, if you don't, you can interrupt me again. Okay, so we wanted to understand how the spatial configuration or the mating range or home range, it doesn't matter how we call this, how this radius was important for a species formation. And something we did, we varied the radius. So here you can see that we varied the radius. We also varied the genome size, and then I explain later why we varied the genome size. But let's focus on the yellow, green, and blue here that are the same genome size with different S values, with different radius. And what we see is that the number of species formed under a larger radius is smaller than those with smaller radius. So here we see that there are fewer species forming under a sympatric scenario, and more species forming under a more parapatric, closer to allopatric scenario that we have in this framework. And we analyze this under at the end of the radiation process. So when the number of species had equilibrated around a value. And after this, we wanted to understand better how these species were related, what were the properties in the phylogenetic trees that we could detect, and if these patterns in the phylogenetic trees could be related somehow with the radius or the geographic mode of speciation that they were they evolved under. And to do this, we had to construct the phylogenetic trees. And this was a great part of our work for a long time. And one of the methods that we can use is recording all speciation and extinctions and everything that happens under this module, which is very computational cost. There is a high computational cost for this. So we can do this just because we have like all individuals and we can follow every event that happens in this model. But still, there is a very high cost to do this, doing this. And yeah, and because we did this, and then we proposed it. Yeah, and then we proposed another method here that records that through a very summarized matrix, we can follow the most recent common ancestor of individuals and then construct the phylogenetic tree. So here is less costly, and then it's easy to make many simulations. By the way, the individual-based simulations are very costly by itself. And then if we need to compute every event and everything, it's more and more costly. So what we did, we used it. It came from from every work in phylogenetic trees. They use many methods. This is one of the methods they already use, but we adapt this for the individual-based model. So now we had the methods to construct the phylogenetic trees, and then we could detect the signatures that were left in these phylogenetic trees by the microevolutionary forces. And when I'm talking about the microevolutionary forces here, I'm talking about the mating range the individuals are using, how is the genetic flow between individuals in the space. And what we observed, and here I'm going to focus again on the yellow, green, and blue, is that when we have the more pedipathic / allopatric mode of speciation, the trees were less accelerated. When we say less accelerated, it means that the phylogenetic trees, they have like long branches. These species are formed in the beginning, and then they stop forming species. So they have this shape in the phylogenetic tree. While the species that were the simulations that were under higher higher radius, we observed that the phylogenetic trees were more tippy, or these phylogenetic trees were more accelerated, meaning that the species took a very long time to form. When they formed, many of them were formed. And we didn't observe, at least in this scenario here, that the spatial configuration was affecting the balancing of this phylogenetic tree. When I'm saying about the balancing, it means, oh, sorry, this appeared somehow. I don't know how to remove it. I mean that when they are balanced, there are as many species in one side of the phylogenetic tree than in the other. When it's unbalanced, it means that one of the branches of the phylogenetic trees gives birth to more species than the other. So it becomes unbalanced. And, we compared this with empirical data that we found in the literature with speciation processes that were adaptive or non-adaptive radiations. When we say adaptive, it's because there was some kind of selection happening during the speciation process. And when we say non-adaptive, there was no force in this process. And we also collect data, and this is not measured, but it was related by the authors, if the genetic flow during the speciation process was low or high. And we related low with the smaller s, and we related high genetic flow with larger s. So the individuals could exchange more genes during the speciation process. And what we observed here is that, for instance, the accelerated trees or the accelerated speciation process were related with those with larger s and larger values of radius. And something that we observed is that most of the phylogenetic trees that were non-adaptive were balanced, while many of the adaptive ones and under low genetic flow were less balanced or unbalanced. And although we cannot say that we understand the geographic mode of speciation and what makes and relate this with the real phylogenetic trees, we have some signatures here